All right, so I'd like to welcome our final speaker of this morning, Dr. Anthony James. Dr. James is a distinguished professor in both the microbiology and molecular genetics department at the School of Medicine at the University of California, Irvine, as well as the molecular biology and biochemistry department at the Ayala School of Biological Sciences, also at UC Irvine. Dr. James is a member of the National Academy of Science. He's a fellow of the Royal Entomological Society, the American Association for the Advancement of Science, the Entomological Society of America, and the American Society of Tropical Medicine and Hygiene. He is the recipient of a Merit Award from the National Institutes of Health and the Atlee Clark Achievement Award for Outstanding Research from the University of California, Irvine School of Medicine. So his research is focused on his research group uses genetics as the basic for synthetic approaches to prevent transmission of mosquito-borne diseases, and he's going to speak with us today about work he's using with gene drives to prevent transmission of malaria.
Well, it's a real pleasure to be invited to speak here, and as you will see, this is going to be very different. All right. The original title was La Zanzara vitruviana, synthetic biology and malaria. And so when it came out saying synthetic biology and malaria, I thought maybe some of you thought I was going to be talking specifically about the parasites, but I'm actually talking about the mosquitoes and the transmission there. And this image will become obvious why we have it here. And this is clearly one of these things where you're standing on the shoulders of giants, all right? This work is made possible by the pioneering efforts of the people that we've heard here, and I guess I'd like to say, coming from LA, we're users of this technology, per se. Okay, great. So, a while back, someone pointed out that most lectures were actually answering questions that hadn't been asked. And I thought, well, maybe I'll change the way I do my lectures. And so there's a question at the top there. And so this helps you follow along what's going on here. So what is malaria? It's an infectious disease. It's an intracellular protozoan. Parasite, what we see here are images of malaria parasites in a human blood smear. Those of you who have taken your basic hematology in humans recognize that these cells should be empty. We see inclusions here which represent the malaria parasites. And this is a diagnostic gold standard for establishing malaria infections. It's mosquito-borne. It's transmitted by the members of the genus Anophelines. And I can see that this looks a lot like one of the phages that we've been seeing, all right? It's a little bit different, a little more complex. It can fly, all right? But it has some of the features that we've seen before. There's a high degree of host specificity, and this is significant here. There are human malaria parasites that only infect humans, and there are mouse malaria parasites that only infect mice. Humans don't get mouse malaria. Mice do not get human malaria, and this will become relevant later on when we talk about the design of effector molecules to interfere with parasite development. There are no free-living forms of the parasite. They either have to be in a human being or they are in the mosquito. You think that might be a tenuous life cycle, but you think about it, there are a lot of mosquitoes, a lot of people, and it works quite well. And the WHO estimates that somewhere around a quarter or a third of a billion people have cases every year, and the number of deaths is actually going down, about 438,000, but we still have a way to go. So how long has malaria been a problem? Well, from the earliest recorded literature, this is pre-PubMed, there are examples of deadly recurrent fevers, which we recognize as being malaria. Hippocrates, a very competent physician, noticed that there was an association of these fevers with a swampy water, and also looked at the fever cycles, which turned out to be diagnostic for the specific parasite species. And we see a first clear association of malaria in history with the recovery of DNA from a malaria epidemic about 450 AD in Lugano, Rome. And throughout this period, there's always been a call for to determine what's going on, and then ultimately in the 20th century, calling for the eradication of this disease. So how do we get to malaria eradication? Well, there's a number of pretty well-described steps to do that start with control of the disease, and these are steps that are defined by the World Health Organization. But the idea here is you get the number of cases down to a certain amount so you have a very low amount of infection every year. With extra effort, you can get to what's called local elimination. And the idea is if you could locally eliminate malaria everywhere in the world, you would have eradication. And so this idea of being able to go from one place to another place, et cetera, achieving control and elimination also will feature later on in some of the discussion that we have here. Now, we have a number of existing tools that work really well and indeed are responsible for the fact that malaria deaths now are probably below 1 million, below 500,000 per year, whereas as short as maybe 15 years ago, they were well up around the area of 2 million. And what we see here are existing tools that work very well. We have bed nets that work very, very well. And I understand Brian Greenwood got the Gardner Award a while back, and he was instrumental in instrumenting this type of control. Historically, we also have what's called indoor residual spraying, which depends upon the behavior of the insect. Insect feeds on blood and then flies to the nearest wall. And if you coat that wall with an insecticide, it will die. And you can actually block transmission that way. Environmental management, where you modify insect breeding sites, also is important, as is the development of drugs. Recent discovery of artemisinin and application in dual drug therapies has had a major impact here, but the parasite is keeping ahead of the drug development. And it's estimated we need a new drug about every 5 years to actually keep up with the pace of drug resistance. Well, if we have these tools, why is malaria still here? Well, there are cases where the current tools have never been applied for geographical, political, economic reasons. There are just some very difficult places in the world to work. We have examples where tools are applied but did not work. We saw the bed nets. Well, they work very well on insects that feed at night on people when they're sleeping. You suppress that malaria and you find that they're outdoor daytime biting mosquitoes, all right? So there's some residues of transmission as a consequence of that. And there are clearly circumstances where the tools used to work but don't work anymore. And in the case of the vectors, this has to do with development of insecticide resistance and also the collapse of public health infrastructures as a consequence of conflict and other political situations. So we think that there's a clear need for better use of the existing tools, but we often also argue that we need a development of new tools. So what are we doing about it? We're looking at novel, what we call genetics-based strategies to prevent malaria transmission. We're talking about genetically engineering mosquitoes. And we have a life cycle here. I told you no free-living forms, either in the human and the insect. Now you see the origin, of course, of Zanzara vitruviana here, the classical image here of Da Vinci for the Vitruvian man. He just drew a man at that time. If he was doing it today, it would probably be a woman as well. And so we're looking at genetically engineered mosquitoes. And we believe that it can be highly effective, species-specific because of the genetic aspect, and cost-effective. So what are the genetic control objectives? Well, the first one is straightforward. Many people would like to see develop and are developing, and that's basically a population suppression tool, where if you're not bitten, you won't get the disease. And this is essentially a genetic analog to an insecticide. And this reduces or eliminates the mosquitoes. But there are other ways of thinking about this, and that is to recognize that it's the pathogen that's actually the target. And we call this now, I guess today, population alteration. It used to be called replacement and modification, but we're looking for less charged words to describe it. So population alteration is coming forward. But here we're changing the ability of a mosquito to actually transmit the pathogens. And the benefits of population alteration are, we think that we can have a sustainable impact on malaria. And this, we don't have enough time to go into arguments about not arguments, but descriptions of sustainability and why this is important. But we think that this technology can provide a sustainable approach here. It leaves behind no empty ecological niche that can be recolonized by either the same species coming back or a new species. With things we won't be able to get into right now, but it's stable in low population densities of mosquitoes, which many of the malaria vectors are. But the significant thing is it allows for a serial approach and resource-constrained control and a consolidation of gains, meaning that if you go to an area and clear it of malaria using this specific technology, you can move to another area to work with some confidence that the area that you freed of malaria will remain so. If you do that today with existing tools and you stop your interventions, it's highly likely that either the mosquitoes will come back or the parasites will come back, get a reemergence of the disease. And so this is that sustainability aspect that I was talking about, the ability to put something in place that will keep that area free of malaria so that you can put your resources somewhere else. All right, so how do we achieve population alteration? I know we're running late, so I'm talking very fast. I apologize. So we'll get to that later. So we're going to engineer mosquitoes with genes that prevent malaria parasite transmission. And this has to deal with the genetics of vector competence. If you think about it, there are a lot of things that feed on blood. I think up here you have black flies. You can think about fleas and mosquitoes. And there are a lot of bloodborne pathogens. We can think about malaria parasites, dengue viruses. We can think about HIV. We can think about hepatitis. And not everything that feeds on blood transmits all these pathogens. And indeed, there are clear host-pathogen interactions where the genetics of the vector and the genetics of the pathogen coincide so that you can actually have the ability of these things to transmit. So it gets even more interesting because if you look within species of vectors that can transmit, you can select for populations that are resistant and some that are susceptible. And if you cross them together, you can start doing genetics. And one of the things that came out of these studies was that in many cases, it was one gene or a small number of genes that had a major impact on whether or not a specific vector could actually transmit a specific pathogen. And we thought, well, wouldn't it be great if we could somehow increase the frequency of those genes in a particular population, and we have a population that could longer transmit. Well, it turns out doing that with naturally occurring genes is going to be very difficult, and so we try to do a synthetic approach. In this context, actually making genes. And then the hypothesis behind this work then becomes that if we could somehow introduce that gene into a population of mosquitoes that would transmit and turn that population from one that was capable of transmitting into one that was not, then we could actually see a reduction in disease and death. All right. So in order to do this, bootstrapping this, we had to define what the major research areas were. The first thing is, could we actually make a mosquito in the laboratory that was resistant to malaria parasites? Having done that, would there be a way to get it out into the field? All right. And then third, what's the field? And you can see here 3 images of places where malarias are transmitted. One is a village in sub-Saharan Africa. Another is a riverside village on the Amazon, and the other is an urban area in India. These are very different. So when we talk about going to the field, there's a lot of complexity there that we also have to think about. Well, how are these genes made and introduced? Once again, this is a synthetic approach. We made a very simple model of a gene where you have a control region, and you know, listening to all these great talks, this sort of molecular biology looks so trivial a bit here. But we have a control region that tells you when and where and how much of something to make. And we use endogenous mosquito promoters to actually drive the expression of our effector genes. And then we have the infector gene, which kills or disables the pathogen. And what we see here are a group of mosquitoes expressing a reporter for a promoter that we're interested in. And these are images of the malaria parasites, the infectious forms. And then finally, of course, we need transgenesis. All right, straightforward sets of tools here. Well, what should we know about mosquitoes? Well, you don't need to know that they're whole metabolous insects, all right? But this is a slide that I've used on many occasions. But they have distinct developmental stages. They're very much in the group of like butterflies where you have a larval form, a pupa, and an adult form. And it's the adult forms that are problematic. But importantly, it's only the adult females that actually feed on blood and are involved in transmission, all right? And the blood is a source of protein for nutrition and egg production. What should we know about malaria parasite life cycles? Well, it's complex, very complex. We have distinct developmental stages in the mosquito host, which are illustrated here, and distinct developmental stages here in the human host. And we think this complexity may lead to some of the reasons why it's so difficult to develop a vaccine. All right, well, what is the experimental system? I'm going to show you a little bit of data and then hurry on to lunch here. One is that we're working on the human malaria parasite, Plasmodium falciparum. This is the most dangerous one and the one responsible for the vast majority of deaths that are seen. We're working on a mosquito called Anopheles stevensii. It is a vector of malaria in the Indian subcontinent and responsible for a vast majority of malaria transmission in southern Asia. What is the strategy? Well, I told you that the malaria parasite has a complex life cycle. When a female mosquito feeds on blood, she takes in specific forms of the parasite that are present in the midgut here. The parasites have to negotiate this environment, get out of the midgut, make their way through the open circulatory system of the insect back to the salivary glands, where they're then transmitted during the next feeding. So a mosquito has to bite twice in order to be involved in the transmission cycle. The first time to pick up the parasites, developmental maturation of the parasite to the mosquito, and then bite a second time to actually transmit. But we looked at this biology and said, well, we can define specific compartments where we could actually interfere with the parasites. The first one being the midgut, where the parasites first come in, And indeed, a lot of the naturally occurring resistance to malaria parasites we see is an inability of the parasites to negotiate this environment. They just don't get out of the midgut for some reason. And then we have the open circulatory system and the salivary glands. So the idea was to look for endogenous mosquito genes that were expressed in these specific compartments and use those as the control regions to actually impact the, or control the effector molecule. And this is some early data for those of you of a certain age. I recognize Affymetrix type data here, defining expression profiles of genes. This is one that's expressed in the midgut here, very highly in the female. Here's one using a PCR analysis. And basically what we're looking at is expression of a gene after a blood meal. Here's a non-blood fed female. Here's a blood fed female. This expression level goes way up. And the same thing for this particular gene here. This is a carboxypeptidase in the gut. This is a vitellogenin gene, which is in the open circulatory system. So these are genes that are turned on very profoundly following mosquito infection here. But it's those control sequences we want to use to drive the expression of our effector molecules. And once again, this image here is to show how we determined that we actually have the effector promoters to do this work. What are the malaria resistance genes? Well, remember I told you that the malaria parasites have a high degree of host specificity for the vertebrate host. So colleagues of ours working on trying to identify antigens for the human vaccine put human parasites into a mouse. Now, any good mouse would not want to get human malaria. It would be an embarrassment, all right? They don't get human malaria. So what they do is they mount a very strong antibody-mediated response and they kill the parasites. What we heard earlier on about monoclonal antibody technology, so colleagues of ours put human parasites into mice, found monoclonal antibodies that reacted with surface antigens of these malaria parasites, and some of them would interact with surface molecules on the parasite that would disable the parasite in the mosquito. They put mosquito stage parasites in the mouse and identified those. So we essentially engineered those by cloning out the variable portions of both the heavy chain and light chain and made a what's called a single chain antibody. And this looks reminiscent of that little simple model that I showed you where we have a control region and effector portion. And the idea is if you're lucky, your single chain antibody will have the same kind of impact on these malaria parasites that the parental IgG molecule did. Now, we made a couple of other modifications. These were mouse genes. We turned them into mosquito genes by doing codon optimization. We actually added an antimicrobial peptide and made something that we call an immunotoxin. We made transgenic mosquitoes, and we made them based on two single-chain antibodies, one that blocks a stage in the midgut and one actually that blocks a stage of the parasites as it moves to the salivary glands. The reason we did this is that early on we were told that, well, you can subject these parasites to immune selection, essentially, and you're going to get resistance. And we said, okay, we'll just put two in. It'll be like a dual drug use thing. We'll put two molecules in. The probability of a parasite having resistance to both of them at the same time would be really low. So we have a dual gene strategy. How are the experiments done? Mosquitoes are fed on an infected blood meal. So this is an artificial feeding here. We give additional meals, and this is a simplification of the experiments that we've done. And then we check and look for parasite numbers. Basically, what we're really looking to see is at the end of this challenge, whether or not we have parasites in the salivary glands that could actually be transmitted onto a human being. And the bottom line is that here's 5 experiments using infection levels that are found in nature. If we look in controls, we see lots of parasites in salivary glands here. 1 is sufficient to cause disease. Okay, so our target has to be 0, but in our transgenics carrying both of these genes, we see no parasites. All right, so this is our phenotype that we want. All right, so this is great. We've been able to take a mosquito in the laboratory, put genes in it, and turn it from one that would normally transmit into one that would not. How do we get this out into the field? Well, there's a couple of ways you can think about this. One is inundated release, which is just to make a whole bunch of our laboratory mosquitoes and start releasing them like crazy. Well, not crazy, but in a rational way, but lots of them. All right, and you just keep doing that, and you keep doing that. And then eventually, your parasite-resistant mosquitoes here, mated with the wild type, if you keep doing that, you will integrate your gene at a high enough frequency that at some point you'll have a population that's all carrying your genes. Can you imagine writing a grant to do this? All right. All right, another one is called reduce and replace. And here and this uses a different technology that we developed, which is a population suppression technology, but you link your effector, your resistance gene to a repression gene, and at the same time you're releasing them, you're killing the population, so you're reducing the size of the wild type population, but you have to continue to reduce, okay? But then, of course, the punchline and why they asked me to come here is gene drive. A technology which, if you have your parasite resistance gene linked to a gene drive system and you made it to wild type, all the progeny that you get out have your gene in it. All right, and we worked for a long time, for Define, for those of you who haven't thought about this. So what is gene drive? Fundamentally, it's a genetic observation that you have preferential inheritance of a specific genotype. And it's been talked about circumventing Mendelian's patterns of inheritance. I have a very deep respect for Father Mendel. So the question of violating his rules, it never comes up. This is just a different genetic phenomenon, all right? The drive system is a final synthetic product that achieves the preferential inheritance. And the drive mechanism is actually the underlying biological drive feature that's incorporated into the system. So the consequences of gene drive are that if you have a Mendelian inheritance here, where you have an animal that's homozygous for big I and big C, you cross them, you get a heterozygote, you outcross it, you get a 50-50 segregation. But in gene drive, you do that same sort of thing, all your progeny carry this I gene in that case. So this is a schematic representation of that. All right, so what are some of the gene drive mechanisms? Well, there are a number of genetic phenomena that people looked at, things that are involved in what we call chromosome mechanics. And here's an underdominant system, a very elaborate genetic system that it will achieve this, and people are still looking at this. There are other things like meiotic drive, and then the gene conversion events. And we've heard the excellent lecture before, this actually leads to this. And of course, factoring in that is the ability to make a double-strand break that will allow this homologous recombination, homology-directed repair. And I'm always going to say, I'm not going to talk about that, but of course that's exactly what I'm going to be talking about. All right. There are a number of other things, infectious-like agents, things like extracellular and intracellular symbionts, viruses, paired transgenesis, transposons, et cetera. But the big thing is the Cas9 CRISPR system for this. All right, and this probably doesn't need any more introduction, so I'm just going to tell you what this system looks like. This is something that we developed now for the mosquito as an autonomous gene drive system to drive itself through a population. This is a genetic construct. It contains the ability to autonomously express Cas9 and guide RNAs. The Cas9 endonuclease is expressed by a promoter that's active in the germline. So the idea is that this will be heritable, and we have ubiquitous expression of the guide RNAs. It carries a dominant marker for DS-red that we can actually monitor where the gene is, and then it has the target gene is one that creates a recessive phenotype so we can actually monitor the targeting. And of course, then we put a cargo on this, which are our dual anti-pathogen effector genes that carry them through. How does it work? Once again, the previous talk explained this quite well. We presume we start as a heterozygous. We can get essentially gene conversion, which is functionally equivalent to the is homology-directed repair. Or we can get mutagenesis, which would be the non-homologous end-join as a consequence of that. What do we see? Well, if we have our gene drive system in males and we outcross it to females, so here's our drive element, these are the wild type loci there. Here are some of the here are some of the data that we had. Where it's pink here, and I apologize for those of you who can't see the red, but these are the pink squares and these are the white squares. What we see is that the vast majority of our progeny have this. So this drive system is working highly well in this system here. And these are from males. They have the normal colored eyes, but if you look very closely, you can see the DS-red, which we can score very easily. And basically, we see there's about a 98% transmission. Contrast that with the 50-50 in Mendelian inheritance. The gene conversion rate is well above 97% or homology-directed repair. If we do the females, so we start with a female here, we see the same kind of drive, and we're looking at the red here. So we see 99% of them actually are carrying this, but now they have white eyes, okay? That indicates the proper targeting, okay? And this is what these look like. Here's some of the white-eyed females, they have some mosaicism associated with it. And it's much easier to see the DS red under those circumstances. So this is for the G2 cross. When we take the female crosses out another generation, though, either using males or females from that initial female cross, we start to see that the conversion breaks down. In fact, the conversion rates actually get very low. In this case, 12% and 19%. So going through the female line, this gene drive appears to be shutting down. So the question is actually what's going on. And it turns out that this has to do with the biology of this insect. All right, this insect makes an egg, like all insects do, which is fertilized by a sperm. So if the gene drive system is coming in from the male, all the machinery for the gene drive system comes in with the male chromosome here, the female chromosomes there. These two chromosomes meet and then segregate into the germline and the somatic tissue. The gene drive system is only active in the germline, so we only see it occurring in the germline cells here. The somatic cells remain unconverted. They don't have that impact on the eye, and therefore these are dark black eyes. And then this is passed along in that manner there, and we get conversion there. However, if we go through the female line, the egg is essentially a test tube full of Cas9 and guide RNAs. So the minute the male sperm comes in there, it is acted upon by the Cas9 CRISPR system, and in the absence of the proximity of the female chromosome template, this actually drives it to the point where we favor the non-homologous end-joint mutations. However, her whole somatic tissue and germline tissue contains mutations then in this particular target gene, and therefore she has white eyes. So what this argues to us is that if we want to maximize the efficacy of our drive system and minimize the recovery of resistant targets, we need to drive it through the male line. We also then looked at the effector molecules after 6 generations, and just through transcriptional levels, these genes were still active. All right, so what are the conclusions? Powering through this, aren't I? Cas9-mediated gene drive works well in the malaria mosquito, Anopheles stevensii. That may not seem like a big deal. It's just a mosquito, but this is a legitimate metazoan organism, and it's not a mouse, but it's, this is good. That's good for us. It's target specific. We saw that these were going to that locus that converted to white eyes. So the vast majority of the circumstances we hear, we don't see off-target effects. Now, we've only looked at several tens of thousands, okay, and obviously we want to be looking at millions, but we think we can get beyond that. It works in the germline of both males and females, but in terms of applications, it will work best in males. It can carry a relatively large cargo. I didn't say when I was describing the size of that gene, but that was 17 kilobases, which is, we imagine it can actually get quite a bit larger, but that was our initial size. The cargo is functional at the transcriptional level, and we believe that strains based on this technology then could have a major role in sustaining malaria control and elimination as part of the eradication agenda. So what will it take to make happen? First of all, there's a scientific realm of sort of, we call these interrogatory realms because we couldn't come up with anything better. But the scientific one, can it be done? Can we make translatable science? And I think we've demonstrated that. We have transmission, transgenesis, effector genes, and we've got some ideas about how the release strategies will actually work. Regulatory then, well, may it be done? We need functioning regulatory systems. This is technology that's being developed for lesser developed countries, all right? So this is not as non-trivial as it sounds. And we need to be able to develop contained experiments before we go to open release from this. And then there's the aspects of community engagement. And I used to say, will it be done? But my colleague said I should make it, should it be done as well, all right? And this has to do with then understanding approval, support, and sustainability. And we need to develop surrogates for what it means to have community consent. And community consent is very different than individual consent. Individual consent is something, every time you go to see a physician, you sign a piece of paper that says you agree to be treated. How do you get that same kind of agreement going to something that has a community-wide application? All right, and I'd like to bring up this example. In the United States, we have fluoridated water, which we've had for many, many, many years. Can you imagine trying to do that today? All right, trying to get fluoridated water instituted as a public health measure in any freely democratic society, I think would be very difficult. So we need to come up with mechanisms for how we're going to get community consent. They're going to be highly specific to the country you're in and require a very deep knowledge of the culture of that country. If you can do that, and it is our job to find places where the answer to all of these is yes, so we can actually do pilot studies. Well, what will it take to happen? We have a lot of industry colleagues here. They're conscious of this idea of a pipeline where you go from discovery to development and delivery. In our field, we talk about drugs that are prophylactic and therapeutic for malaria, insecticides and vaccines. And the point is that there are well-established pathways for doing this, making progress from one step to another. For genetic-based vector control, we're just in the process of developing that. And some people would argue that you have the foxes in the hen houses because the people who are helping to do this are people who are also advocates of the technology. Having said that, we also know what the liabilities are, and so we're working to establish trust with people to develop these types of technologies. Along that line, working with the WHO, we've adopted this idea of phase trials, where we have phases where we're working with laboratory cage experiments. And the idea here is that the insects must meet specific design parameters as they move from the laboratory stages to phase trials, which could be contained cages in the field or geologically confined on islands, or some countries actually may require you to do both. And after that, we would like to go to open field stages, and following that with successful meeting of the goals, we would actually be in releases that have an impact on epidemiology. Each stage requires regulatory oversight, community engagement, and communications. Now, I put this picture up here because I want to emphasize that I do not believe that our technology alone is going to be the single thing that's going to allow malaria eradication. All right? This is a tool and a large number of tools that are needed to achieve this goal. And these other tools include things like insecticides, drugs, monitoring, vaccines, public health infrastructure, personal protection, you can read around the wheel here, community involvement, diagnostic tools, et cetera. We need all of this before we're actually going to be able to get to eradication. So please don't go away thinking that I've described to you a silver bullet. Ours is part of this approach to eradicating malaria, the ability to sustainably control, eliminate malaria in specific regions where this technology will have an application. Almost got us back on time. All right. So I'd like to acknowledge the people who will work on this. These are folks in my lab. Neola Jasinskian was the first person ever to transform mosquitoes and continues to work in my lab because she can still do it very well. We got some exciting things going on with that. Fred Gould's a modeler at North Carolina State who's helping us. Valentino Gantz and Ethan Beer are at San Diego and developed this technology for Drosophila as a way to rapidly produce homozygous mutants in Drosophila. But when they shared their data with us, it was immediately clear to me that this was a gene-drive system, and we put it to a practical use. It's likely that I will have to leave before the discussion, so I will hang around for some minutes afterwards here. If you have specific questions, you can come up and ask me. Thank you very much.